My name's Mark Coyne. I'm with the Department of Plant and Soil Sciences. And Chris Teutsch and Ray Smith are two of the nicest people I know. So I can't imagine what the KCA did to annoy them so much that they would invite me to come and speak to you today, because I've been putting students to sleep in soil microbiology for the past 27 years. I can only think it's a conspiracy that they got me up here so that they look really good. But we're going to talk about the soil system, and it's a good that you've left a little bit of space in the splash zone here, because when I talk about soil microbiology, I get really excited and I tend to spit just a little bit, so you're fortunate there. Now, first thing I want to have you do is give yourself a round of applause. Because if you do your job well, what you're doing is exactly what Charles Kellogg said is important for the soil environment. That you are making that soil alive, and you are preserving that soil for future generations. What you're doing is of great benefit to all of us. So what I want to do today is remind you of 4 things that you're doing that's helping to preserve that soil biology. I want to talk about the setting of the soil environment. I want to talk about the organisms that are there, the players in that soil environment. Talk a little bit about what they're doing, and then talk about some management issues that specifically affect the soil microbial population and the other bigger organisms that are in the soil environment. So the setting, the players, their roles, and the management. Okay? So let's get started. I am a soil microbiologist by training, a soil scientist by training. I am contractually obligated to show you a soil horizon in any talk that I give. Welcome to my world. My world is a thin layer of the Earth's environment that is about 2 feet deep, 60 centimeters. It's got this funny green stuff on top of it, but what I'm really interested in is the stuff below. This nice, rich layer of organic matter accumulation. And then a little bit farther down, a zone of biological activity. And then, of course, we have leaching of nutrients, and we've got clay movement. But if you look closely over here and over there, you see that there are actually roots penetrating much deeper into that soil environment. And technically speaking, that too is part of what I'm interested in. The activity that occurs around those plant roots. So what we're going to focus on today is those things that are happening in that very thin slice of the soil environment that we can directly manage. Now, what you may see, again, this funny green stuff on the top of the surface. But what I see are 5 overlapping different types of microbial habitats. The drillosphere is a unique environment around the pore, around the burrow that an earthworm is going to make in a pasture. The detritosphere is that layer of detritus and litter and organic matter that's lying on the surface of the soil. The porosphere is exactly what it is. It's the pore space in the soil environment. The rhizosphere is that environment around the plant root that influences and is influenced by the microbial population within the soil environment. And finally, the aggretosphere is that environment that the soil aggregates form when clays and silts and sands coalesce and make larger and larger aggregates. And if you look inside those aggregates, what you find is they're just absolutely full of pore space. And it's in that pore space, depending on how big those pores are, that you find all this microbial life, you find all this insect life, you find all of this worm life. It's just an environment that's teeming with life. What kind of life? Well, lots of different things. And if you really had to ask me what my job description was supposed to be, I might point you to this bit right here. I'm supposed to come up with creative ideas to control and direct these microbial groups for some useful end. Whether that useful end is to produce more plants, whether that useful end is to purify air and water, or whether that useful end is to find some novel type of organism or novel product that those organisms make that can be beneficial. Okay, so it's teeming with life, a myriad of organisms. What are the sorts of things that exist in the soil environment? Let's talk about the players. And we're going to go from big to small in this sequence. Big ones are going to be the earthworms. These are the things that you can see. This is your classical earthworm. Smaller than that, but still pretty big, are the Microarthropods, the big insects. And that would be an example of a big insect. It's actually a tropical insect, but it's nice and colorful, but you can find big ones on your own as well. Smaller than that are the Microarthropods, the small insects. And this would be a representative sample of the different types of Microarthropods that you might see if you were to extract them from the soil environment. Smaller than that, we have the nematodes, or what you might have heard of, the roundworms. And this is an example of a nematode. It's actually one of the most numerous animals in the soil environment. Smaller than a nematode, but now a single-celled organism would be the protozoa. And this would be an example of a protozoa. Smaller than the protozoa, in some cases, are the fungi. One end of the fungi you can see, the fruiting body, a mushroom. That's the fungi. But the working end, the business end of fungi in the soil environment are really the hyphae. They're the ones that are most important in terms of transformations in the soil environment, in terms of putting that soil structure together. Smaller than the fungi are the bacteria. And this would be an example of bacteria. Smaller than the bacteria are the viruses. Now, I've got good news and bad news for you about viruses. The good news is if you're exposed to this kind of virus, it's called a bacteriophage, you don't have to worry because it only infects bacteria. On the other hand, you might be infected by this type of bacteria. Well, don't worry about that one either. That's tobacco mosaic virus. And unless you're a tobacco plant, you're not going to be infected by tobacco mosaic virus. But how many of you feel achy and sneezy and a little bit feverish? Well, that's good because everyone around you should be moving away very quickly right now. That's influenza, and that can infect people. But most of the viruses in the soil environment affect the soil microbial population. Is there anything smaller than the virus that's actually active in the soil environment? Well, yes, there is. It's the enzymes. You can actually have enzymes in the soil environment that remain active even though there isn't any living organism around them. And I can't actually remember if this is the model of the enzyme urease, which is responsible for breaking urea down into ammonia, or if it's alcohol dehydrogenase, the enzyme that's responsible for breaking ethanol into other byproducts. I guess that depends whether or not this is a drinking crowd or not in terms of what enzyme you're most concerned with. So, big organisms all the way down to the molecular level. Hold this in your hand, 5 centimeters or so. This is at the molecular scale. An enormous range of size of the biology and the living things that are in that soil environment. And it's an enormous population. If we look at the number of organisms per square meter of the soil surface, we go from maybe 100 snails and slugs all the way to billions and billions of bacteria in that environment. Huge numbers. And small organisms with lots of numbers, even if they don't weigh much, ultimately add up to a lot of mass. So in a typical square yard or cubic yard of soil, you might have 2 to 3 pounds of living organisms. That adds up. It ends up being about 1,300 pounds per acre of living organisms. And I put this slide in to ask a question: what is the typical stocking rate in a normal pasture? And I'm not talking about mob grazing or anything like that. What would be a typical stocking rate? Not seeing any response there. 1,000 pounds of animal per acre. Well, if you think about what's above ground then in terms of the number of animals, there's about 8 cows worth of biological activity below that soil surface. There's more stuff living underneath the ground than above the ground. And again, I wasn't quite sure from Chris whether or not he said the 65 cattlemen was an overestimate of the number or an underestimate. Depends on how big he thought the typical cattleman was. So think about it. You're managing an above-ground biology, or you see an above-ground biology, but you have to consider below ground, there's 8 times as much stuff going on that could potentially be managed. Now, the thing about the size of an organism, the smaller you are, the greater the diversity of organisms you have, so that if we go from our large macrofauna, things like earthworms and ants and termites, we've got, several thousand species, and we know what a lot of them are. Most of them are described. But as we get smaller and smaller and smaller, we find that there are lots more organisms or types of organisms present, but we know less and less and less about them. And they're all working together. This is a diagram of a typical food web in the soil environment. Your plants, your photosynthesizers are making carbon and organic matter for the organisms present. You have decomposers, things like bacteria and fungi and nematodes that are breaking down that organic matter and turning it into nutrients. And then you have protozoa and nematodes and microarthropods that shred that material, but also consume the bacteria and the fungi and turn them into nutrients and more insects. And then you have bigger level predators feeding on the smaller level predators, and you have even bigger organisms feeding on those. Now, there's something fundamentally wrong with this diagram. Because if it just continues going like this, ultimately all you're going to end up with is a bunch of groundhogs and moles in your pasture. It's a cycle. And so let me just say that, there we go. I am a scientist at the university. I am contractually obligated to make things more complicated than they need to be, which is why this diagram is up here. That if you think about it, our resource base is at organic matter. The plants and the roots and things like that. The fungi, actinomycetes, and bacteria are consuming that. That carbon and that energy is flowing up through nematodes and rotifers and microarthropods. And ending up in this pool of litter transformers and other insects. Now, some of that organic matter goes right to litter transformers or right to our, what we're going to call our soil engineers, our earthworms, that mix it with the soil environment. But then ultimately, that material all flows back into the resource base to get recycled again. So in a good nutrient cycle, a good managed system, you're keeping your inputs, you're keeping your material within that cycle and continually cycling and rotating them through that system all the time. And you want to ask yourself a couple of questions. If you increase your resource base, if you increase the amount of plants or roots or organic matter, what's that going to do to the network of organisms that you're able to support in that soil? And if you have a larger group of organisms present, if you increase that carbon flow, that energy flow, that nutrient flow, what does that do for nutrient availability in terms of making more plants? So the way the system is supposed to work is more or less like this. You have your plants, and as your plants decompose, and their roots decompose, it's fragmented by a group of organisms that turn it into smaller bits that are mineralized, that are turned into soluble nutrients by the organisms in the soil environment. And now soluble nutrients get taken back up by the plants and turned into more plants that die, that get fragmented, and so it continually cycles. So your plants are harvesting carbon from the atmosphere, water from the soil, nutrients from the soil, and putting that back into the system to support and sustain a whole group of other types of organisms. And if you have a lot of organisms, a lot of diversity, that's great because it gives you a lot more different ways to go from point A to point B. Use this analogy. And Lexington's an easy city for us to use this analogy. Would you rather be in a city that had lots of roads that were open or lots of roads that were under construction? Particularly if you wanted to bug out of town very quickly to avoid the snow that's coming. The fewer roads you have, the less opportunity you have to get anywhere. The greater the number of different paths that you can take in the mineralization and the decomposition and the transformation of things in soil, the better it is for the whole system overall. That's why the NRCS says diversity, 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 diversity. This is why. The more diverse you are, the more opportunities you have to make things happen. So who's doing what in the soil environment? We basically divide our organisms into three major groups. We have our soil ecosystem engineers. These are the big people. We have our biological regulators. These are middlemen doing lots of transformation. And then we have our chemical engineers and microbial decomposers. And they're not completely mutually exclusive in terms of what they do, but they really essentially have functionally different roles in terms of how important and what they do in the soil environment. Ecosystem engineers, you can see these guys. You can pick them up out of the soil. You can extract them. These are the earthworms, the millipedes, the centipedes, the beetles, caterpillars, scorpions. They build big pores in the soil environment. They redistribute soil particles. They mix organic matter in the soil environment. Charles Darwin said they're like the best plow you ever had. They're moving tons of soil from the soil surface, subsurface, all the way up onto the surface of the soil. So earthworms, enchytraeids or potworms, a bunch of different types of earthworms. So you can divide them into three big basic groups. Epigeic, these are the wrigglers, the things that are in that detritus or litter layer. The endogeics, these are the dirt eaters. These go back and forth in the soil environment, consuming organic matter in the soil. And the anasics, these would be the things like the night crawlers and the earthworms that burrow up and down and bring soil organic matter into their burrows. Some of them are really big. This one is about 1.5 meters long. If you've ever had an earthworm that big, we typically don't have them in the US. And they can do some visible topographic changes in the environments in which they're found. So this is a habitat in Africa where there's been so much worm activity that it's made ridges and valleys in that particular soil environment. That's why they're called ecosystem engineers. They're changing the physical environment of the soil. Now this looks like an unusual field, and it's actually thousands of years old. It was created by Native Americans in South America to deal with flooded environments in this floodplain. But when they left or abandoned it, things like ants and other macroinvertebrates moved in and persisted in maintaining those environments, even after you stopped having agricultural activity here. So the macrofauna, those ecosystem engineers, are capable of dramatic changes in the soil environment. The insects, they may be transient residents, they may be permanent residents of the soil environment, but you can see what they do. Termites, you just hope they're not in your house, but they're pretty good for the soil environment. Burying beetles, ants and ant nests, isopods or pill bugs, centipedes, millipedes, and of course, we have a whole group of ground nesting solitary bees. And so this brings up one of the interesting points, or an important point to realize, is that when you add chemicals to the soil environment, you've got to be conscious, is there anything else? Is there collateral damage to useful insects within that particular environment? And a good example would be these ground nesting solitary bees, because they're important in pollination as well as the typical honeybee that you see. Biological regulators, these are organisms that regulate the populations of other smaller organisms. They're preying on them, they're feeding on them. So the things like protozoa and small invertebrates, nematodes, springtails, and mites. And one of the most important things about these organisms is that they're pretty messy eaters. And by messy eaters and an ecological scale, it means that they leave lots of nutrients behind. And the nutrients that they leave behind when they're consuming the microorganisms means that plants can take up those excess nutrients and grow a little bit better. So, they're actually, nematodes and protozoa are actually very good for forage development, for plant development, because of the nutrients they leave behind. Some good or other examples of these organisms, the meso and the microfauna, are the nematodes, and this is an example of a nematode that's actually being consumed by fungi in the soil environment. Everything in the soil environment has something else that's going to feed on it. You can have herbivorous nematodes. They'll have a stylet like a hypodermic needle that'll puncture a root and allow them to suck out the nutrients. But you also have these bactivorous nematodes that feed on bacteria, consume bacteria. There are 4 groups of protozoa in the soil environment. Here are 3 of them. You have testate and non-testate protozoa, ciliated protozoa, and flagellated protozoa, depending on how they move in the soil environment. Again, these are consuming organic matter. These are consuming bacteria. They're cycling those nutrients around for other organisms to use. Somewhat bigger than the protozoa and the nematodes are these microarthropods. You've got mites and springtails and pseudoscorpions and protura, rotifers, my favorites, the tardigrade, a water bear. But they help to fragment material. They help to consume fungi and bacteria. And again, messy eaters, they're cycling those nutrients around. They're making those nutrients available for other organisms in the soil environment. Stepping down, the chemical engineers, my favorite group. They're regulating the energy flow and the carbon flow in the soil environment. They're cycling nutrients. Bacteria, archaea, fungi, some protozoa. Enormous diversity with minimal size. Start with the fungi. This is what you typically are going to see. Lots of different types of fruiting bodies. And 2 very important types of fungi are the types that form symbioses with plants in the soil environment. Really, 90% of all the plants in the soil environment have a type of fungi, either an ectomycorrhizal fungi or an endomycorrhizal fungi, that infects them and helps those plants get nutrients, particularly phosphorus. The ectomycorrhizae form this sheath around things like trees. The endomycorrhizae actually are going to infect individual cells and plant roots and make these dendritic, these tree-like structures called arbuscles that allow them to transfer nutrients to the plant and get nutrients from the plant in order for them to grow. Again, we see this and they're pretty, but the working end of a fungi is the hyphal network that they put in the soil environment, because that's helping to put compounds into the soil environment that make aggregates form, as well as to get nutrients out of that soil environment. The bacteria and actinomycetes, we have bacilli, we have spirilli, we have cocci, and they can be individual, or they can form long chains or packets and things like that. They can have flagella that allow them to move. They can be nitrogen-fixing and photosynthetic, like the cyanobacteria. They can be hyphal-like. They can look like fungi, even though these are prokaryotic organisms, meaning they don't have a cell nucleus, but they have a huge variety of different morphological forms, and this is the group that right after it rains and right after you till up or dig up some soil, if you take a handful of soil and you put it up to your nose and it just smells good, that's a compound called geosmin. And that compound is made by actinomycetes. On the other hand, if you take up a handful of soil and it smells like somebody died in that soil, that's also due to microorganisms, but it really reflects that that is a soil that's probably too wet or probably has too much nitrogen in it for really efficient, useful nutrient transformation. You can have spore-forming bacteria. So lots of different physiology, lots of different types of bacteria in the environment. So how do you manage soil? But you have billions of organisms in the soil environment. You have enormous diversity. How do you manage it? Again, I'm a scientist. I am contractually obligated to show you a slide with far too many numbers on it for your viewing pleasure. But how can you manage? Well, one of the very best things that you can do is add organic matter. And this is an example where they've taken a reclaimed mine site or reclaimed site and they've added biosolids, either 7% or 15% biosolids. Now, what happens to the organisms in soil? And we're really just looking at the largest organisms in soil. What happens when you add organic matter for that population? Well, generally speaking, if you look at this circle here, you find that if you add organic matter, you get more organisms. That's a good thing. But organic matter has variable effects on these organisms and the types of organisms that are affected. So, for example, with Collembola, the springtails, their population steadily goes up the more organic matter you add. On the other hand, this particular group, as you add more organic matter, the number of these organisms goes down. And this particular group, the isopods, the pill bugs, the roly-polies, you add a little bit of organic matter, the numbers go up. You add more organic matter, the numbers go down. So anyone who tells you that there is a magic ratio or number of organisms in the soil environment better have a really good proof or demonstration that that's true, because it is such a complicated environment. And which of these organisms do you care to manage? Because obviously, if you manage it one way for one organism, you're going to change the management for another organism. Imagine trying to manage 10 trillion different types of organisms in the soil environment on a routine basis. So think about it. What's easier to manage? Soil biology or manage the soil environment on a day-to-day basis? If it were me, and I wanted to add more carbon to soil, because I know adding carbon to soil is a pretty good thing. It generally is very good for diversity. Well, I might just simply grow more forage. Well, how do I grow more forage? Well, you might pick the right forage, you might pick the right pH, you might pick the right fertility, but I'm a soil microbiologist. Fortunately, there are other speakers on the program who actually know how to pick the right forage, how to pick the right pH, and how to pick the right fertility so that you can grow more forage. So I'm not going to try and talk about that. I will leave that to Chris and John to say how you're going to do that. You can manage animals as well, and that is actually going to have an effect on the microorganisms. There are three major grazing effects: trampling, defoliation, and defecation that are going to influence microbial populations. Now, trampling tends to be bad. It tends to be bad because it leads to soil compaction, and that means that you have less pore space. And the organisms in soil like to have lots of pore space, big pore space. So it reduces that, so overall it's going to diminish the number of macro, micro, and mesofauna. Defoliation, trampling is also going to reduce the amount of litter. And remember that these are organisms that require litter, require organic matter in order to grow. So if you reduce the amount of litter, you're going to negatively affect them. Defoliation. Well, you're reducing the amount of litter, but one of the things about defoliation, it might stimulate some root exudation, and root exudation is good for microorganisms, fungi, and bacteria, so that might actually end up increasing the organisms. So you can manage microbial populations to change by the amount of your grazing strategy. Patchy defecation, where you have nitrogen and readily available organic matter from manure deposits coming into the soil environment, that's a good thing. You're adding organic matter to soil. You're adding nitrogen to soil. You're adding phosphorus to soil. But if it's patchy, there's only going to be some environments that are affected. So how can you make more of that environment better influenced by the amount of defecation that occurs? Well, that's something you can manage. So I'm going to give you Coyne's law. And it's a heretical law for most soil microbiologists, but I think it's a good law. And it's basically the lazy microbiologist law. Remember that sort of quasi-job description that I said, that I need to use constructive imagination to devise means to control and direct all the organisms in soil to some useful end. Well, I would rather somebody else do it for me because I'm a lazy microbiologist. And so here's Coyne's law. Make your agronomist friends, make that physical and chemical environment appropriate with sound management. And if you do that, you don't have to worry about the ecosystem appropriate biology. That's going to follow good management. That if you build it, if you build that useful management system, then the soil properties are going to follow that, and the soil organisms are going to follow that. And appropriate ratios, even though those ratios of different organisms may vary from time to time and place to place, they're going to be appropriate for that time and for that place. If you build it and develop those organisms, they will come and they will thrive. I'll leave it to our other speakers to talk about how do you do that appropriate management, and if you pay attention to them, then you've taken care of your soil biology for the most part. I have a number of individuals, a number of groups that supported me, my college, USDA, the NRCS. Microbiologists don't steal, we recycle. So the pictures that you've seen have come from the USDA or a book called The Atlas of Soil Biodiversity. And if you have any questions, I would be happy to answer them.